welcome to the second annual webinar offered by Sustainable SWD Management SCRI project, sponsored by USDA NIFA Specialty Crop Research Initiative project. Today, we are going to talk about advances in behavior-based tactics for management of spotted wing Drosophila. Just for the intro, spotted wing Drosophila is a vinegar fly of Asian origin. It was first detected in the mainland US in 2008 in California, and since then has spread throughout major small fruit producing regions in the United States. It has caused serious damages there are several reasons for it. But one of the foremost reasons is that this fly has a really short life cycle. It can complete all life cycle from egg laying to adult emergence in 8 to 10 days at 25 degrees centigrade, which means it can go through several generations and build up populations to cause a lot of damage in all small fruits. The key hosts include blueberries, blackberries, raspberries, strawberries, cherries, and many other fruits that this fly can cause damage to. In a survey, the way this fly works is that female has this very unique ovipositor, which is sclerotized, which means it's hardened, and it has serrations on it that work like a saw that female uses to puncture the skin of otherwise healthy fruit to lay egg inside the fruit. The larva comes out within 1 to 3 days, and it starts feeding inside the fruit. And within very short period of time, it basically changes a healthy, normal, marketable fruit into unmarketable. That leads to fruit rejections at the packing houses, and in most fresh fruit markets, the tolerance levels are very, very low, almost zero tolerance in the fresh fruit market. In a survey, the estimates were made for the annual crop losses to be about $718 million and increased management costs of $129 million. Again, these damages were estimated back in 2014, 2015. Current damage values may be much higher, and we can safely say that this is a $1 billion pest for our small food industry in the US. So this is a group of researchers, when this fly invaded in the US, a group of researchers from several major land-grant universities got together to start doing research on it. Initially, we had 2 phases of SCRI projects that addressed the urgency of the situation and developed management programs to control it as soon as possible. Then this is our 3rd phase of SCRI, where our goal has switched from addressing the immediate concern to developing more long-term management strategies to control this pest. The main goal of our SCRI project is to develop and deliver systems-based integrated management programs to berry and cherry growers that are cost-effective and environmentally sustainable for long-term management of SWD in the US and worldwide. Specific objectives of our project are listed here: implementation of best management practices for sustainable management of SWD in collaboration with grower influencers. The second one is develop economic-based decision aid tools to support identification and implementation of profit-maximizing SWD management strategies. Then evaluate sustainable alternatives to long-term SWD management, which include behavioral control that we will cover what we have learned so far today, and then biological control that our team is working on, that we will provide an update in our next webinar. Assess and reduce the risk of insecticide resistance development, and develop and disseminate actionable recommendations that enable producers to optimize pest management decisions and evaluate their impact. As I mentioned, this is a large team of researchers from several major land-grant universities in USDA. As you can see here, University of Georgia, North Carolina State University, Oregon State University, Michigan State, UC Davis, UC Berkeley, Cornell University, Rutgers University, University of Maine, Washington State University, and USDA. This is a list of this project participants. And if within our project team, we do have objective leads and objective teams, and today's seminar content will be presented by and other contributors are part of our what we call SWD behavioral control team, which is Cesar Saona from Rutgers University, Ash Sayal, myself from University of Georgia, Yvonne Walton from Oregon State, Rufus Isaacs from Michigan State, Philip Fanning from University of Maine, Greg Loeb from Cornell University, Elizabeth Beers from Washington State University, and Frank Zalem from University of California, Davis. Today's presentations content were contributed, all of these researchers contributed to the presentation, and 4 of us will be presenting today. This point on, I will hand the control over to Cesar Rodriguez Saona to present the next part of the presentation.
Thank you, Ash. So in this webinar, we will discuss new behavior-based control technologies that our team is evaluating to manage spotted wing Drosophila. So I will start by discussing attract and kill strategies. Then Dr. Von Walton will be talking about a technology called decoy or food-grade gum. And we will end with Dr. Greg Loeb talking about repellents for spotted wing Drosophila. So our team is testing new technologies that combine attractive cues, feeding stimulants and insecticides to attract and kill spotted wing Drosophila, and also to increase the efficacy of insecticides. So one attract and kill technology that our team has been testing is Hook SWD. This product is a waxy matrix that slowly releases attractants. And more recently, we have been testing a product called Combi Protect that is commercialized as an insect bait for spotted wing Drosophila that contains feeding stimulants to increase the efficacy of insecticides. So Hook SWD is an attract and kill formulation based on a technology called Splat. Splat is a waxy matrix that allows for slow releases of volatiles. And this formulation contains attractants, phagostimulants like sugars, and an insecticide. And it can be applied both manually or mechanically, for example, with the use of customized ATVs. So early on, we conducted field trials where we treated small plots 2-3 times during fruit ripening. And also we had control plots that received no Hook SWD. And then we took fruit samples from these plots and look for SWD emergence in the lab. So these studies show that Hook SWD can reduce infestation by about 60 to 90% during the first week after the application. But also we show that efficacy is reduced after 2 weeks after the application. We also have done some trials in commercial farms using the customized ATV to apply Hook SWD to the lower parts of bushes. And these applications were done either weekly or every other week. So in these studies, we found that infestation of fruit in these farms was low, but still we could reduce SWD infestation both by weekly applications and by applying every other week by 20 to 30% in days where the pest pressure was high. So more recently, we tested whether the efficacy of Hook SWD breaks down under high fly populations of spotted wing Drosophila. We did a study in cages where we released different fly densities. We released 0, 20, 40, 60, and 80 flies inside the cages. And we either treated bushes inside the cages with hook applied to the leaf or to the bark. And also we had bushes that had no hook SWD. So we found that hook SWD is more effective under low fly populations, and it tends to break down when the densities of the populations increases. So a new formulation that has recently been developed by the same company that produces hook SWD, ISCA Technologies, is ACTRA SWD. This formulation contains the attractants and the phagostimulants, but do not have an insecticide. Thus, it works as an adjuvant. So a grower would need to mix the insecticide with these formulations. So the company has developed two formulations of this product, ACTRA SWD OR1 and ACTRA SWD TD. So I'm going to refer to them as OR1 and TD. The hook is the ACTRA SWD OR1 plus spinosad. So we conducted studies in the laboratory to determine the attractiveness of spotted wing Drosophila to both of these ACTRA formulations. And we also evaluated the effects of the spotted wing Drosophila sex, the age, and the physiological status on the response to ACTRA SWD formulations. So for this, we used two choice arenas where flies were given a choice. For example, in this case, they were given a choice between blueberries and the TD formulation. So the arena was provided with a water source, and also moisturized cotton pads. There was also a small mesh opening for air circulation. And we released 20 insects in the arena. And then we looked at the response after 24 hours after the release and calculated the % response to each of the choices. So for example, here, the flies were more responding to TD than the control. So in these choice arenas, we tested the response of flies to ACTRA formulations in the presence and absence of host fruits. We selected blackberries, strawberries, raspberries, and blueberries, and as a choice. And for the hook SWD that is shown here, we found that others from all the fruit types tested were significantly more attracted than the hook SWD. And this was a very similar response when we looked at the OR1 formulation. However, for the TD formulation, only raspberries were more attractive than this formulation. So these results suggest that when a fruit host is present in the field, the TD formulation will be more competitive in attracting the insects than the other formulations tested. So we also tested the influence of spotted wing Drosophila sex and age on its attraction to ACTRA formulations when they were in the presence of blueberries as an alternative choice. So we found that age did not seem to have a strong effect on spotted wing Drosophila attraction to the ACTRA formulations. Also, we found the OR1 formulation was not influenced by the sex. So you can see that males and females responded similarly to that formulation. However, for the TD formulation, males were more attracted to the TD formulation than was the case of females. We also conducted bioassays with these two ACTRA formulations to identify the influence of female mating status and also the effect of fruit ripeness on the level of attraction to the formulations for both TD and OR1. We found that for both formulations, fruit ripeness did not influence the female attraction to the ACTRA formulations. So you can see either ripe or unripe, overripe fruits were equally attractive. However, female mating status significantly influenced the attraction of ACTRA formulations. And compared to unmated females, mated females were more attracted to the fruit, the blueberry fruit, over both ACTRA formulations. So more recently, we have been testing a new product called Combi Protect that is being sold as an insect bait. This product works as a feeding stimulant and can be used as an adjuvant. So growers can mix this product in their tanks with an insecticide. And in the last year, in 2021, we tested 4 different treatments. So we looked at untreated controls. We looked at the delegate and entrust at the full rate. We looked at the delegate and entrust at the full rate plus the Combi Protect, and also the delegate and entrust at half rate with the Combi Protect. So our team conducted studies in large field cages in both high bush and low bush blueberries, and also in raspberries. And these studies were done in Oregon, New Jersey, Maine, and New York. So we released about 100 flies inside these cages, and then we sampled fruit and also evaluated the level of fruit infestation, and also recorded adult emergence. So in Oregon, high bush blueberries, we found that delegate alone or delegate alone with the Combi Protect, both at the full rate and also delegate at the half rate with the Combi Protect, provided good protection against the spotted wing Drosophila compared to the untreated controls. So these results were similar for New Jersey. Also, we found that Combi Protect, both at the high, the Combi Protect with delegate, both at the high and the half, at the full and the half rates, provided equal control as the delegate at the full rate compared to the controls. Also in Maine, low bush blueberries, we found the same results. Both the delegate at the full and the half rate with the Combi Protect provided similar control as the delegate with a full rate compared to the controls. And in New York, we found similar results. So this year, what we did was we conducted additional semi-field experiments where we treated individual bushes. Individual bushes with an insecticide at either half rate or full rate with and without the Combi Protect. So we also had bushes that were untreated, and then we collected foliage and fruits from these bushes and exposed the spotted wing Drosophila to the treated foliage and the treated fruit in these containers. We looked at, then we measured mortality of the flies, the number of eggs that they laid in the fruit, and also the number of adults that emerged. So these are the results from Georgia. These are the results for adult mortality, where 4 insecticides were tested. This included Fosmet, Zeta, Cypermethrin, malathion, and Spinotherum delegate. And you can see that in all cases, here you have the untreated controls. In yellow, you have the half dose. The dashed ones is the half dose with the CombiProtect. Red is the full dose, and the dashed red is the full dose with the CombiProtect. And pretty much the half rate and the full rates of the insecticides with and without the CombiProtect had similar results, showed similar results. So these are the results for egg counts. And again, both rates of all the insecticides with and without the CombiProtect had similar effects on spotted wing Drosophila oviposition. And these are the results for adult emergence. In general, both rates of the insecticides with and without the CombiProtect also had similar effects on spotted wing Drosophila adult emergence. So in Oregon blueberries, they tested 2 organic insecticides, Entrust and Grandivo. And Entrust at both the full rate and the half rate with and without the CombiProtect increased spotted wing Drosophila mortality. For Grandivo, the addition of CombiProtect, as you can see here and here, to either the full rate and half rate, at least numerically, increased the efficacy of the insecticide at both rates. Entrust and Grandivo had no effect on oviposition, as you can see here. However, Entrust at both rates, the full and the half rate, with and without the CombiProtect, reduced spotted wing Drosophila emergence. And for Grandivo, addition of CombiProtect tended to reduce spotted wing Drosophila emergence, as you can see here, compared to here for the full rate and here half rate with the CombiProtect compared to the half rate without the CombiProtect. So in conclusion, we are finding that attract and kill strategies show promise in reducing the spotted wing Drosophila infestation, and this can potentially reduce insecticide use. However, further studies are needed in large-scale commercial fields. We are also finding that the efficacy of these strategies can be influenced by insect sex, the sex of spotted wing Drosophila, the physiological status of the flies, and also the fly population. We are also finding that the efficacy can be influenced by the presence of fruit, indicating that they might not work as a standalone control strategy. And currently, both ACTRA and CombiProtect are going through registration in the US. So now, Dr. Von Walton will continue with his presentation.
Okay, I just wanted to talk a little bit about something different that has an impact on our technologies here itself as well. And in this case, I'd like to talk a little bit about eliosomes. And if you look at all these seeds here, and these are not insects, these are plant seeds. If you look at the nice cream colored shapes on those seeds, those are what's called eliosomes. And if you look at the function of eliosomes, it is to attract insects. And that is kind of the thing that we're wanting to do in some cases, to attract insects so that we can use that behavior to protect us against our crops. When we look at eliosomes, this is an evolution that's taken place more than 22,000 times all over the world in 6 continents. So why don't we use a technology very similar to those? If you look at eliosomes itself, it's got a lot of fats in it, it's got a lot of metabolites coming from those, including really, really important fatty acids. And so it's the fatty acids that results in the attraction of the insect. And so when we looked at our attract and kill, or our attractors, our decoy, we used this very same principle to develop it. And so the goal for this technology was to develop something where that lasts much longer than 1 week. So if we think back of those seeds can last for years under the soil until fire occurs, and then those seeds germinate. When we look at fruits can last, depending on the cultivar, for a month or so. And we want something that lasts in the field for more than just 1 week, maybe something closer to 3 weeks if we can. We also want this to be as attractive as fruit, just like those eliosomes. We don't want to apply those compounds directly onto fruit. And then lastly, in terms of regulation, we want to make sure that these compounds are so harmless that they can be classified as minimum risk insecticides. And so we started this progress years ago during our second grant for spotted wing Drosophila under Dr. Barak. We started looking at attract and kill, and Cesar just shared some of those results with you from their lab. This is a formulated spray that needed to, at least in Oregon, we applied it onto the non-fruiting zone. However, back in that stage, there were some shortcomings to that technology, and we wanted to try and solve that in some ways. So we also looked together with Dr. Tracy Lesky at visual cues. We used the tracticidal swears. We saw some preliminary positive results. However, there were some difficulties in terms of cost for those tracticidal swears, as well as in our system where growers would harvest, and in some cases harvest these swears together with their fruit, which is definitely not an ideal type of situation. During our earlier work on beneficials, we also started looking at fruit and to use fruit to attract spotted wing Drosophila. And we used these fruit in these little sentinel traps to create that alternative media for spotted wing Drosophila. However, the problem with that is that it doesn't last very long. And so we needed to create something that, as I said, lasts longer than a 1 week period, that is as attractive as fruit, not directly applied onto fruit. And so the problem with these fruit is that you need to change them every single week, which is really, really problematic. It's useful to show spotted wing Drosophila and parasitoid population levels. However, it's very, very labor intensive itself. And so during that initial foreign exploration period, we very quickly learned that collecting fruit as opposed to placing sentinel traps is the way to best monitor for spotted wing Drosophila and potentially attract those spotted wing Drosophila in. And so when you look at the life cycle of spotted wing Drosophila, as we see in this picture here, this insect can occur within the fruit for lengthy periods of time, and fruit is really, really competitive to attract spotted wing Drosophila. And we need to find something that is very similar in terms of longevity in the field itself. So the first step that we did was to start identifying volatiles. And in this case, this is work that was published a few years back, identifying those volatiles. And if you notice those 7 volatiles that we're looking at here, these are the exact, in some cases, the exact same volatiles as you find in those eliosomes. We're showing that these volatiles are highly attractive, irrespective of mating status, irrespective of fruit. And the key thing here was to now put those volatiles, use plant material and put those volatiles into those dispensers so that we can be just as attractive as the fruit itself would be. We did this firstly by placing the this tablet. As you can see here, this is a 2 gram 0.06 ounce tablet that is a very, very powerful package. You can see the flies aggregating on those here in the field. You have these dispensers, which is put into the irrigation line. Nothing is sprayed onto the fruit, and then no residues on those fruit itself as well. A lot of work has been done on this technology over the last 5 years, and the newest data is from a commercial field here in Oregon conducted by Dr. Jaina Lee. This is a big trial. These are trials that are conducted over 1 acre blocks, where we had our decoy, we had our buffer blocks and control blocks, 5 replicates of those very late in the season. Here you can see Dr. Q Park placing those dispensers. placing those dispensers are about $7 an acre for time to place them. This is a drip irrigated field. And what Dr. Lee did is she went and looked at egg and larval counts. And generally speaking, the data that she generated from that showed, if you look at egg counts compared to larval counts in the fruit, kind of interesting here is when you look at the egg counts, it is about a decimal higher, about a magnitude higher compared to larval counts. But generally speaking, when we're looking at our control, eggs versus decoy, about a 50% reduction of egg and larvae in both cases. So you'll note here that this is not significant. This trial was done over a 21-day period with one application of decoy during that. Just to kind of give you an idea of how this product works in commercial fields, when growers are using pesticides as well, I'm going to show you the next few slides here. This is a slide where there is absolutely no pesticides that's being sprayed, which gives you an idea of the type of control level. So the question here is this compatible with insecticides? And the answer to that is yes, it is. So you're looking here at 6 different locations, 2 different fruit, and the periods of these trials ranged from 40 days up to 73 days. Our decoy was applied every 21 days. And in this case, when you're looking at the blue line compared to the red line, or the orange line in this case, we're finding approximately 50% better fruit pack out rates. This is a commercial scenario. So just looking a little bit more into the mechanics of this technology, when we're looking at or assuming approximately 50% savings in fruit quality, and we model that, on the left-hand side, we have our controls. On the right-hand side here, we have our gum. We're finding that we're having much lower egg counts, lower larval counts, and then eventually lower adults emerging from those. Taking it a little bit further, you would say to me, well, that is just an academic question. Let's see how this plays out in the real world. So if we look at that modeling perspective where we're reducing egg laying using this decoy, we're finding, and this is over a 49-day period here in Oregon, finding that our grower standard, which is green there, where a grower is spraying 3 pesticides per week. So we're looking in the range of about 20 pesticides sprayed during that period. Compare that to the decoy, we're finding a significant reduction compared to the blue dotted line there in terms of the untreated control. If we compare that with our modeling, we're seeing very, very similar outputs. And so the question here is, how does this fit in with future technologies such as biological control? And so here you have a slide on the top left here where we have no treatments. If we're looking at an insecticide gum combination, as I just showed you in some of those earlier slides, we get a reduction in damage, and this is over a 3-month period. However, if we make some assumptions of adequate biological control together with a sustainable technology such as the gum, maybe something such as the Actra, something like a Combi Protect, we're getting a very, very good result. Now you'd say this is modeling, and I agree with you. This is modeling. No models are perfect, but we would like to think that they are useful sometimes. We're seeing that we're moving closer to IPM. We're moving closer to sustainable management here, and this is real world. So I have a few final concluding remarks here. One of those is that this technology is a technology that was developed in our lab, and there is a conflict of interest here because I'm presenting this data. This is a technology that have now been licensed to a commercial company. It is registered in several states. It's usable. So there is that potential conflict of interest, and I just wanted to state that. This work has been trialed in 8 countries. It has been independently verified by multiple scientists over a period of 5 years. We're finding about 50% reduced damage in the field itself, whether you use pesticide or not. The technology itself lasts about 21 days. It can stretch to 28 days. It is compatible with conventional pesticides. You have no residues added onto your fruit. And then this is a minimum risk insecticide or pesticide, depending on how you would like to look at that. This technology is now, as I said earlier, licensed to a large multinational company, and it has been used in thousands of acres in the last 2 years. And that's it from my side. Thank you so much.
Hi, everyone. So thanks, Cesar and Vaughn. I'm going to take over now and provide an update on the progress the team has been making on repellents. And in this case, I'm going to focus in on work coming out of Cesar's lab at Rutgers, my lab here at Cornell, and work out of Vaughn's lab at Oregon State. But I also want to acknowledge that many people that have worked on all of these projects we've talked about today, from postdocs, grad students, undergrads, technicians, collaborators within and outside of our team. And it's really taken a large army here to accumulate the kind of information we're presenting, hopefully leading towards improved sustainable management. So I'll focus on repellents. And I like this definition from one of the godfathers of entomology and insect physiology, Dave Thier, who defines repellents as any substance that elicits an avoidance reaction in an organism. Kind of straightforward, something that a lot of us would understand. Most of us have experience with repellents in terms of preventing or reducing biting flies from attacking us. And we know from those experiences that things like DEET can be really effective. And as this cartoon indicates, if you forget to put it on, you recognize that how well it does work. Repellents in agricultural systems, especially crop agricultural systems, have not had as much success, I would say, as in trying to repel biting flies. And there's some various reasons and challenges for that I'll come to in a second. But first I wanted to define a little bit better how repellents can work in terms of retarding the attraction to things like berries that spotted wing is interested in. A true repellent, this is what work based on a review paper from a scientist named Delattre. The way we think of these true repellents is that the fly encounters usually an odor. It could occur at contact, but encounters an odor before they reach the target, and they move away from it. They move directly away from it. They don't like it in some way. So we refer to these as true repellents. If they land on the foliage or the fruit and something irritates them, that might get the same reaction of moving away, and refer to those as contact irritancies. Another way that repellents can work really is to mask the target host so that the fly has a hard time finding it. This can involve odor masking, and there's probably several ways this could occur, but the idea is that they have a hard time finding it, the target, or some cases you can work with visual masking. And finally, a repellent might work after contact, where there's some compound on the foliage or the fruit that discourages, in some cases, feeding, or in our case with spotted wing, oviposition. Okay, so what are the challenges developed in repellents that our team has been addressing? First of all is, well, where do we find sources of repellents? Where do we look to find things that might behaviorally affect spotted wing Drosophila in acting like a repellent? And here's some areas that we've looked at. Plant products, botanicals, such as rosemary oil, could be acting and have shown some activity against flies in terms of repellents. Microbes, various microbes, fungi and bacteria produce odors that you might expect to be repellent. And we've done some work on one of these things called mushroom alcohol, 1-octantriol. And then I just had list this serendipity. Like sometimes we have come across pretty powerful repellents by luck looking for something else. So these are sort of the different ways, and I'll talk about examples of each of these in a second. Another important concern with anything you're going to spray on or near a crop is safety, both in terms of human safety, and the environment, non-target effects, things like that. So we have to take that into consideration for some of these repellents. A repellent that it makes people sick is not gonna, it's gonna be more challenging to use than one that is food safe, for example. Another challenge in progressing in the development of a repellent is how do we deploy it at scale? You know, the scale that's relevant to growers. And of course, in all of these is the aspect of cost. Okay, I want to start out with some research that has been going on in Cesar's lab related to odors coming from fermented apple juice. This is work that he and his colleagues at USDA have been doing. And one of the things they discovered in looking at some of these odors, potentially they think they were looking for attractants, but found that some of them were quite repellent. In particular, a focus on methyl benzoate, a compound and a series of other compounds that actually resemble in chemical formula the insecticide I mentioned that's good against biting flies, DEET. So they looked at some of these classes of compounds similar to DEET for anti-insecticide activity for spotted wing. And here's an assay they did looking at oviposition, and they found that in fact this methoxybenzoate product that they're quite interested in worked similarly in reducing egg laying as another another alcohol or benzyl ring product compound produced by bacteria, geosmin, a known repellent for spotted wing. So that looked very promising. Continuing this work, they've looked at average adult emergence for fruit that's been treated with this methyl benzoate, and again found significant reductions comparable to geosmin. So they're very interested in developing this further. Cesar had mentioned to me earlier that they are working with potential companies to kind of put it into a commercialization track in order to be used and purchased by growers. They're not there yet, but that's the direction this work is going with this compound, methyl benzoate. Okay, so they kind of discovered that in some ways serendipitously with while looking at these odors from fermented apple juice. In the next example out of Cesar lab I want to talk about is odors from this fruit infecting pathogen, anthracnose, on blueberries, and it gets on other fruits as well. And they reasoned that if fruit infested with this fungus would not be very desirable for spotted wing, and that perhaps spotted wing would be adapted to identify that at a distance using odors produced associated with anthracnose. And in fact, that's work they were able to show this is the case in behavioral trials where they asked flies to make a choice between anthracnose fungus being reared on a media compared to on untreated fruit, and they definitely do that. Similarly, they collected anthracnose infected fruit from the field and found significant aversion to that by flies. But interestingly, if they took kind of a solution from this colony and filtered out the conidia and different spores, they lose the effect. Clearly, or pretty concretely indicating that it's something to do with the fungal spores that's creating this effect. They've looked at different other aspects of response, number of eggs laid and number of adults emerge, and they get pretty much the same effect, a suppressive effect due to this anthracnose on fruit or in colony. So what are the odors? And they've now carried out really interesting research on the odors being produced by fruit that's infected by anthracnose, and found up to 14 compounds that are produced at a significantly higher rate compared to uninfected fruit. And moreover, some of these compounds, 9 of them in particular, are quite repellent or reduce attractiveness to spotted wing. This is this assay here on the bottom left. And then when they look at these, this assay on the bottom left was conducted in a multiple choice kind of arena. But when they look at pairwise comparisons, they found that at least 2 of these compounds greatly reduced attractiveness, this compound N and compound M, greater than 90%. So they're very much excited about exploring these compounds as potential repellents that we can get commercialized. Similarly, with these compounds N and M, when they do attraction studies there, they work as well, if not better, than other compounds we've identified as repellents like octanol, geosmin, and 2-pentylfuran. So very encouraging results that they're continue to pursue. Now, in our lab, we've been working on a couple of different things. We've initially did work on a couple of micro-produced odors that had been found in Drosophila melanogaster to be repellent, 1-octanol, a mushroom alcohol, and this benzyl ring geosmin. Both of these were repellent in the lab. 1-octanol was quite repellent in the field, both in small and in moderate scale. However, this mushroom alcohol doesn't smell very good, and also at high levels has some toxicity issues for mammals. So we kind of stopped pursuing it because we just didn't think it would ever have a chance of getting registered. We have been working on a different fungus that affects fruit that we've obtained pretty similar results to Cesar. This is Botrytis cinerea, or gray mold. And then we've been looking at another compound called 2-pentylfuran. Let me give you a quick summary of the work here. For Botrytis, like Cesar found, we found that when we put in an attractive fruit, but include a fruit with or an odor associated with Botrytis, flies don't like it, and they'll choose something different that's just fruit only. And we also found that Botrytis reduces oviposition, reduces survivorship, and flies that do come out of fruit with Botrytis are significantly smaller, both males and females. So there's some promise here. We also found that these odors seem to act at lower concentrations than octanol for the similar kind of reduction in attractiveness. We're not as far as Cesar, though, in the sense that right now we have some ideas of what these active odors are, but we're still working on it. And this is work I'm doing with my collaborator at USDA ARS, Dr. Dong Cha. 2-pentylfuran is an interesting example of serendipity. This is another case where we were looking at the odors produced from whole wheat bread dough and looking for things to make the current lure more attractive. And what we found is a number of compounds that actually reduce the attractiveness of attractive fruits. And in particular, the most significant reduction occurred with this compound 2-pentylfuran. Now, we're really interested in this compound because we knew that when we looked things up that it's already registered as a food additive. So it has already gone through a number of safety tests. So we thought, well, this provides some potential for, again, taking it down that road towards commercialization. We've tested this in the field in small kind of scale experiments. Where under very high pressure with raspberry fruit, we see over a 50%, 60% reduction in egg-laying over short time periods. Here we're releasing 2-pentylfuran, which is a highly volatile compound, out of these little plastic sachets. We decided to scale this up using sort of modest size plots of raspberry, again under fall conditions where the pressure is very high, where we put out different densities of these sachets to create kind of a cloud of 2-pentylfuran around the planting. And the results were somewhat discouraging, even at the highest release rates, where we had something like 36 of these sachets releasing at a given rate. We did see a significant reduction compared to our control, but it was not a large reduction. So we're still searching for better ways, again, to deploy 2-pentylfuran that improves its activity under realistic conditions. We are combining our efforts with a pest control company or a pest management company, SCRI Biologicals, the company that also makes one of the lures that people often use for adult monitoring. They're also working with these puffer systems, which you can control the amount of material released, how frequently it's released, how much of it's released, and during what part of the day it's released. So we've been working with puffers releasing 2-pentylfuran, and this is research being done by a postdoc in my lab, Benita Srestha. And Benita conducted field trials, a number of different field trials this past summer. I'm just showing you one of the results during the fall, again, under very high pressure systems, where we had multiple puffers releasing at every 1 minute 8 milligrams of 2-pentylfuran compared to zones in these plantings where they didn't have pentylfuran released. And we see a very slight, not even statistically significant reduction. Again, unfortunately not particularly promising as we're trying to move this down towards commercialization. We're continuing this work, looking to find ways to release 2-pentylfuran that is more effective at a cost-effective rate. And that's kind of ongoing work, but that's where we stand with 2-pentylfuran. Now, I wanted to finish up by some results out of Vaughn's lab, which is really taking it to the field at field scale. This is work he's been doing on plant-derived kind of food-safe products that you can spray out with a normal sprayer. And this is some research he was doing in blueberries in Oregon this past year, releasing these plant-based materials at 60 gallons an acre at 0.075% rate, and looking at infestation, egg and larval counts at 6 and 9 days after application. The material that they're spraying had no apparent effect on the blueberries. You can see the foliage and the fruit look good. However, when they collected ripe fruit, they found obvious signs of infestation. When they took that into the lab, they found significant reductions at 6 days and 9 days of number of eggs per berry on spotted wing, clearly showing a deterrent or repellent effect. So that's really pretty neat at the scale they conducted this. So some sort of summary notes from the work they've been doing, that it seems relatively effective at low rates, has this longevity that's not that different, at least in some of our insecticides, especially organic insecticides. So it's kind of comparable to organic treatments, and it could be used as an organic alternative. It's based on food-grade plant-based materials, so it's safe, non-toxic, and they don't see any phytotoxic effects. The status of this product is that it's in a position where it can be commercialized, and that's kind of where it's at. It hasn't been commercialized yet as a sort of product available to growers, but I think this is an example of where this might add to our toolkit, and something where a company could take this on and make some money and provide some benefit. Okay, so I think I want to kind of finish up here in terms of the work on repellents. I do believe to find better and more effective repellents, we need a better understanding of mechanisms, and so that's an area of active research. I also think that it's true to say that repellents are not likely to be the silver bullet like they are for biting flies, that we're going to need to combine them with other approaches in order to be able to exploit their benefits in a commercial setting. Of course, we need to find repellents that are safe and have minimal non-target effects and are cost effective. Okay, that's the end of my slides, and I want to turn it back over to I think Ash to kind of wrap up our webinar.
All right, thanks Cesar, Vaughn, and Greg for summarizing the work done by our SWD behavioral control team. So basically, the take-home messages from work that we have been doing so far on behavioral control is that behavior-based strategies may help reduce SWD infestations and insecticide used once implemented properly. They are safe and compatible with the other IPM approaches, primarily biological control, which is also part of our project's team's work that we will be summarizing in our next webinar. And this behavioral strategies are not likely to be effective standalone based on what we have learned so far. They will need to be implemented in combination with other control strategies, including biological control, as well as chemical control when needed. And also, we obviously need to determine cost effectiveness of these strategies, and they need to be cost effective and need to be incorporated as appropriately to the systems that they will be implemented in. So at this point, I would like to show you the resources, the online resources that our project team has created. There are several websites that we have created shown on this slide. And the most primary source that we have is our project website, swdmanagement.org. We do post updates from outputs from our project at that website, and also a copy of this a recording of this webinar will be posted on that on this website next week. So if you missed part of it, or you have some other friends or colleagues or neighbors who would like to watch this webinar, it will be available, accessible at this project website. At this point, I would like to thank the sponsor of this Sustainable SWD Management Project, which is USDA National Institute of Food and Agriculture Specialty Crop Research Initiative under award number 2020-51-181-32140. And also there are several other agencies and grower stakeholder groups that have provided this supplemental support to enable us to complete the work that we have done. And also their support is ongoing as we are continuing to make progress on different fronts of this project to leading to sustainable SWD management. And also, as a last thing, I would like to request all of the attendees to take a few minutes, it's not very long, to fill out this survey. Here is a link to the survey. And also you can use this, scan this QR code using your phone cameras to access this survey. Please fill this out. It will help us tremendously in terms of evaluating this webinar content and also making improvement as we organize, as we do more research on SWD management and present those the results to you in other webinars in the future. Slides from this webinar and the recording, as I mentioned earlier, will be made available to all registered attendees and will also be posted on YouTube as well as our project website, swdmanagement.org. At this point, I would like to thank each and every one of you for taking time to join us for this webinar, and I will open the floor for our questions. Please feel free to type in your questions in the Q&A, as you can see, Q&A box, if you have any questions, and we'll be glad to answer. Q&A tab is, if you go in different tabs section, is somewhere in the middle.
You've got a number of folks that have definitely found it, and you have questions that are queuing up for you. There is a request at some point to send out the information sources you had before. Am I right in thinking that swdmanagement.org is the place to go for pretty much all the links that you had there?
That is correct. Most of the outputs from this project will be posted on that, including these slides on that website.
So I'll go ahead and read off the questions that I see. There's been a whole bunch of questions throughout. The ones that haven't been answered, is there any further work going on with exclusion netting? Hopefully cost effective for small organic growers.
That is a very good question. And that exclusion netting specifically is not part of this project, but there's a work going on in multiple labs that under a different project that will be summarized separately.
Hey, Ash, can I add a little bit there that in addition to the OREI, we've been working on it in New York, including working with Miguel Gomez, our economics person for this SCRI, and we are looking at developing economic models for exclusion netting, kind of trying to bring together costs of support systems and netting and the benefits from terms of reduced infestations. And I think we'll be making progress over that in the next year as well as some of these other programs that are looking at it.
Okay.
Thanks, Greg. Appreciate it. Joe, next question.
Is there a timing or application chart being developed for regional application purposes? How do people know when to do what?
That's again, very good question. And for that, we do have some project-wide outputs, but the most reliable source would be your local state specialists. Please contact your local state entomologists who work with small fruits and SWD to get more specific information on application timings and order and season-long programs, which we develop as part of this project as well. But we distribute through the local specialists and your local extension systems.
Okay. Someone is looking for the evaluation link. I've dropped it in the chat again. If you click that, or Ash will bring up the QR code later. you can get back to that. So someone has interested in comments concerning potential for repellents in mitigating development of insecticide-resistant SWD populations. Do you care to discuss that?
Joe, can you read out the question again, please?
I would love to, but someone marked it answered, so I'm going to grab it real quick. They're looking for your comments concerning potential for repellents in mitigating development of insecticide resistance for SWD populations. So you've got these great repellents. How can that be used?
Different.
Yeah.
Yeah, it's a different mode of action. It was me, sorry, I typed an answer in there. So it's definitely a different mode of action, and so it will delay. Resistance is not something that can be eliminated. It'll delay resistance. So we need to use all these tools to manage or delay resistance.
Again, that's the resistance management is $1 million question. And the whole point in our this project is to work on alternative strategies that can be used as part of the management programs to minimize the overall use of insecticides. And that is the best way to manage resistance, is to minimize the number of times an insecticide is applied, and also the modes of actions that are used during one season. So the best thing is to try to not repeat the same insecticides if you can. If you need more spray applications, just rotate between different modes of actions, and also implement other strategies, biological control as much as possible, and behavioral control and other cultural strategies if they are applicable to your system.
We have another question. What do you think about the effectiveness of mass trapping SWD with an apple cider vinegar lure or other types of lures?
That is a good question. We actually did investigate mass trapping as part of another project in the past. That was not part of this project.
Caesar looks like he's ready to jump on that one.
No, I go ahead, Ash. Because we also did try it in the past, but go ahead. Yes,
but the thing is that mass trapping impact on the system depends on the population density of flies. It works up to a certain point. If your pressure is too high, you will still need to supplement that with the insecticide applications to knock the populations down, and then use those soft strategies such as mass trapping. Again, mass trapping outside on the borders, and also using the most effective attractants. Apple cider vinegar is not the most attractive substance to use for attractant to use for mass trapping. We do have other products based on our group's previous work. We do have published different products that are much more effective. And now, actually, there are a number of products available commercially that are much more effective than apple cider vinegar that could be used for mass trapping if that is a desire. But it will work only at moderate to low pressures. High pressures, you will need to supplement with insecticide sprays.
Also, to add to that, what we found was the traps that we use are not that effective at actually capturing the flies inside the trap. So what you have is that usually when you have a trap, it's attracting the flies to the area. So what we found was, in many cases, we were finding higher infestation in the bushes where the traps were located. So unless we find like a better way to capture flies and kill them, mass trapping, I don't think is going to be very feasible.
Can I add one other thing, is that in closed systems, in enclosed systems like netted systems, maybe, and I'm just speculating a bit here, I don't have a lot of data, some of these mass trapping techniques might work a little better where there's not very much movement inside. And if you can just clear out the flies from the enclosed area, you can maybe have more success. Having a better attractant, something that's more attractive than the fruit, or at least as attractive, is pretty important there.
We have a couple of questions and I'm going to combine them together because people are asking about not only the trap, but the lure. What's the most effective trap and lure that's being used right now for monitoring? Is it still apple cider vinegar? Is it the trace day traps? What's being used and what's best?
Cesar, you want to take it?
Yeah, that we have been testing more recently the use of sticky cards, red sticky cards, baited with either the Sentry lure or the Tressa lure, and that makes trapping easier instead of like having to go through a liquid trap and having to count, like, you know, a lot of flies. And we, in our case, in New Jersey, we only count males that are easier to spot in this on the sticky cards. So we're trying to make it easier for growers to monitor by using these kind of traps that are, you know, easier to set up and also to check in the field.
Also, I would like to add here, the Hannah Barak just posted that our last year's webinar covers this subject of monitoring and trapping in real detail. And the link Hannah posted in the Q&A window. Please take a look at that and use that to view the webinar. That also was posted on our project website, swdmanagement.org. Please watch that that webinar for more detailed information on monitoring and using those traps.
I've also dropped that same link into the chat because everybody can't quite see all the fun stuff in the Q&A spot. And Hannah is on it with the links. She's also got the link to SWD Management. Here's where all the webinars are. Yeah, I think there's definitely what Ash and the others have done. SWD Management is the place to go if you want any of this information.
All right, next question, Joe.
There are currently no additional questions. So we'll make call. If anyone has any more questions, we're 11 minutes over, but that's fine. We're here and it's good to make sure we've got these answers done.
All right, yeah, last call for any questions you may have. If you prefer to actually speak up, we can give one opportunity to open up your mic. Just raise your hand or type in the chat or somewhere.
Mass release of parasitoids. How's that coming?
That is outstanding question. And our project team has worked really hard first to get the permits and now to establish regional insectaries in most of the labs that are participating in this project. And most labs have already established good colonies and have started releases at select sites in their respective states. If you need more information about those parasitoid releases and the status of those, and if you are a farmer and you would like your farm to be used as an initial test release site, please let your local SWD research specialist know, who, as I showed in the project team, when you review this webinar, look at our project team names and contact your local researcher and get information from them. We'll be glad to work with you to use your site as a release site. We always look for good sites and good collaborators to do those releases. And next year, our plan is to provide a detailed update on biological control or using exotic parasitoids in our webinar next year. So stay tuned for that. And at this point, I'd like to thank each and every one of you again for taking time to participate in this webinar. And we again will get back to providing information on our next on our research progress every year. We have this project website. Please visit that for more information. If you have more questions related to this webinar or anything related to SWD, please feel free to email me at ashsile@uga.edu, and I'll be glad to direct those questions to the appropriate person, the specialists who can answer those questions. Again, thank you very much. We really appreciate it. And stay tuned for more progress on swdmanagement.org. Thank you very much.
Everyone have a great day.